Alright guys, in the last video we had talked about signal transduction pathways, which is represented by the picture that you see in front of you. We had kind of reviewed a little bit about the reception of the signal molecule, known as the ligand or the first messenger, and how that message is then relayed to the second messengers, which then activate all of these sequential proteins in turn. And we discussed the need for this, the need for the fact that we have to have multiple proteins activated in this pathway. Number one, so we can amplify the signal if we need to, increase the amount of product that's going to be created, and also so we can regulate it and fine-tune it and things of that nature, okay? And so we discussed these things called protein kinases, which are like on switches for this cascade of activation. We talked about phosphatase enzymes, which are the off switches for this, represented in this picture by the small pp in a purple oval, okay, and how they shut off these active molecules. Both of these on and off switches are needed to properly regulate the amount of product or cell response that is created at the end. And then we talked about this, and we've mentioned this idea of the cellular response over and over and over again, and I've been very ambiguous about it and haven't given you any details about it because I knew we were going to be doing this video right here. Okay, so today's video is going to be addressing this cellular response that I've been mentioning for the past three videos, and we're gonna discuss some details regarding it. Now, we're not going to get overly detailed because there are hundreds of thousands of different responses that you could have because of an external signal that's received by a cell. So we're going to talk in some generality, but we are going to offer a little bit more detail as a result of that. Okay? So cell signaling leads to regulation of cytoplasmic activities or transcription. That is the main idea that I look to get across for this particular chapter, is the fact that when we receive a signal, or when our cells receive a signal from an external source, we will usually have one of two general responses. It can be cytoplasmic, meaning this response happens in the cytoplasm, or it can be something that is nuclear, which happens inside of the nucleus, okay? And so this response is generically called the output response. So the cytoplasmic or nuclear responses are going to give you different types of reactions, okay? They're not necessarily chemical reactions, but reactions inside the cell, different types of things happening. For instance, cytoplasmic responses oftentimes happen to regulate some kind of metabolic function. Maybe they do actually regulate chemical reactions, okay? Nuclear responses are going to be ones that maybe turn on a gene and create some kind of protein product, okay? So by the end of, the reception of the signal, the transduction through the cytoplasm, you're eventually going to get this this regulation of many different types of cellular activities, okay? So as we work our way through this last section of video, I'm going to discuss a very, a very detailed cytoplasmic response, which you will see in the test. There will be an item on the test about this, so make sure that you maybe bookmark this little part of the video, okay? And we will talk also about nuclear responses. All right. So the cytoplasmic response that you see in front of you looks a bit daunting and intimidating, but it's okay. You've seen all of this stuff before, okay? And I'll try to go back and show you a picture of kind of where you've seen this before, okay? So the cytoplasmic responses regulate the activity of enzymes in the cytoplasm. Okay, so that's kind of the main goal. Now, there's plenty of different pathways that we could talk about, and most of those pathways are far too difficult and detailed for us to really get into in this class. So I've decided to just pick one, the one that the book kind of talks about, and it's one that you guys have all experienced at some point. So it might be a little bit easier to remember as a result of that. Okay, so the one we're going to talk about here is the binding of epinephrine. Okay, so epinephrine, if you guys know, is what we affectionately call adrenaline. Okay, adrenaline causes you to either fight or flight, right? It is responsible for you either fighting some attacker or, fighting something so that you stay alive, or running from that something so that you can stay alive. So, I like to use the example of meeting a bear in the woods, right? What would you do? It's not a little fun little teddy bear. It's a giant 9-foot tall, 700-pound grizzly bear. Okay, it's giant. This thing's huge. And so you're walking along and you see this giant grizzly bear, and it does not come up and nuzzle you. It comes up and tries to swipe at you, and all of a sudden you feel something inside of your body. It might be a little pee that's run down your leg, but it could be something else too. And that something is probably the adrenaline rush that you've got. Okay, now that adrenaline rush is going to give you the energy to allow you to either fight the bear to the death, which you'll probably lose, but sometimes that's the last resort, or run away and get away from the bear as fast as you can. It provides you the energy to do those types of things. Now, also think about these times you see in the news where a mother, is in a car wreck. I've heard of something like this, right? Mother's in a car wreck, and the car is rolled over, and the car is laying on the child, right? And then the mother lifts this car up like the Incredible Hulk. Normally, under normal circumstances, maybe that's not possible. The mother's not capable of that particular feat of strength. But because there's this massive adrenaline rush, she can lift it up just enough so the kid can get out of the car. Okay, so those are things that we have to try to relate to as we discuss this particular example, because I think it will start to make a little bit more sense when you discover what goes behind that epinephrine or adrenaline rush, okay? And how we can relate it back to this cell signaling cycle. So I'm going to go ahead and get the little things where I can draw on here. Okay. I'm going to change this so I can draw a little bit better. All right. All right, so my face is right in front of you, but I'm going to draw on this thing a little bit better. All right, so we look up at the top here, and this diagram that you see in front of you has all of the different phases of the signal transduction pathway. It's got the reception phase up here. It's got the transduction phase here in kind of aquamarine blue, and then the response phase here in yellow. Okay, and go to red. Okay, so we notice at the very beginning in the receptor phase, the G protein linked receptor is the receptor that is going to be used in this particular pathway, and we have epinephrine binding to it. Okay, and we start this off with one molecule of epinephrine in one particular cell at one particular point on the cell membrane. Okay, so one molecule of epinephrine binds to the G protein linked receptor. Okay, it starts with that. Now the next thing we see is that our G protein becomes activated. So try to think back to these diagrams we saw with G protein linked receptors, right? The first thing to become active was the G protein linked receptor itself. The next thing to become active was the G protein, right? Now what you notice here is that all of a sudden we go from one molecule of stuff, okay, one molecule of epinephrine, to all of a sudden 100 molecules of G protein are activated. Okay, so right now we can start to see the importance of the signal transduction pathway and how it has developed over time. Next, we have 100 molecules of active G protein are going to activate 100 molecules of adenylyl cyclase. And that's what it's telling us here, 10 to the 2. Okay, so basically we look at a one-for-one activation. G protein, one molecule of G protein will go and activate one molecule of adenylyl cyclase. Okay, which is, if you go back to the diagrams showing G protein linked receptors, you'll see adenylyl cyclase. In some of the pictures it's not labeled, in other pictures and animations it is labeled. Okay, so go back and check this stuff out. I will visit that picture again to help you kind of get a visual representation of actually what's going on. Okay, now 100 molecules of adenylyl cyclase is going to activate, I think that's how that works. You add 2 zeros, right? 10,000 molecules of cyclic AMP. Cyclic AMP is our second messenger. Okay, second messenger. All right, it's the finger that pushes the dominoes. Okay, now we're going to push the dominoes. Cyclic AMP. There's 10,000 fingers going to push 10,000 dominoes of protein kinase A. Okay, so again, we have a 1-for-1 activation. 100 molecules of cyclic AMP is going to activate, 10,000 molecules of cyclic AMP is going to activate 10,000 molecules of protein kinase A. So at this point right here, we will activate the signal transduction pathway, okay? The cascade of sequential activations that we talked about in the previous video, okay? All right, so 10,000 molecules of active protein kinase A are going to activate 100,000 molecules of phosphorylase kinase, and that's another type of protein kinase, okay? They just give it a different name. All right, on we move. 100,000 molecules of phosphorylase kinase is going to activate 1 million molecules of glycogen phosphorylase. Now, these last two, you don't necessarily need to memorize the names of these things, okay? Just know that they are proteins in this cascade, okay? But when you take some time and you look at the name of this one in particular, glycogen phosphorylase, it'll actually kind of tell you what it does, okay? So at this point here, we have 1 million molecules of this final protein that have been activated, okay? And our signal transduction pathway at this point is kind of done. Now we're going to use these 1 million molecules to elicit some kind of cellular response, okay? So we have started with 1 molecule of epinephrine, and we have amplified that signal into 1 million molecules of something known as glycogen phosphorylase. And so what glycogen phosphorylase does is it breaks apart glycogen. Okay, I'm going to give you a second. Maybe if you could, you pause the video and try to remember where you've heard glycogen. I'll give you a hint. It was in chapter 5, that all-important chapter 5. And try to remember what glycogen is and what its function is. So pause the video and see if you can't remember that, or maybe even go back and look it up and see where we're going with this, okay? All right, so glycogen phosphorylase, what that molecule actually does is it takes this molecule glycogen and it breaks it apart, okay? Glycogen is made up of thousands of monomers, right? And those monomers oftentimes are glucose molecules or other sugar monomers, monosaccharides, okay? So it's thousands of those things put together. And what we as animals do is we store our sugar in that form, and we store it in the liver, okay? So glycogen is the storage form of our sugar molecules, all right? It's where we put it when we don't need all of it. Now we need it. We've seen a bear. You've wet yourself. You either have to run away or poke the bear's eyes out and rip his jaws apart and win the fight. It's life or death, right? And so now you need all that stored up sugar. So these 1 million molecules of glycogen phosphorylase are going to then give you one, let's see if I can do this math. I'm going to take 1 million and add 2 more zeros. Okay, so my commas are off. 100 million molecules of glucose 1-phosphate. Glucose 1-phosphate is a derivative of glucose. It is a molecule that your mitochondria can break down and give you massive amounts of ATP in a very, very short amount of time. So you now have 100 million molecules of a glucose derivative that will allow you to make massive amounts of energy. Now can you understand why you feel like you could fight a bear or lift a car? Or when you get that adrenaline rush, that feeling of what you're experiencing is a massive energy rush. And the reason is because you've just liberated 100 million molecules of glucose in one cell. That's one cell, okay? And when you've liberated those 100 million molecules, you make far more ATP. For each molecule of glucose, we get, it's like 38 ATP or something like that. And so you've got 3.8 billion molecules of ATP from one cell. I mean, you are getting massive amounts of ATP based on this signal transduction pathway. And it is that massive energy surge that allows you to sometimes do superhuman things or feel as if you can do superhuman things, okay? And what this does, this feeling, it allows your body to utilize oxygen at a higher efficiency. It shuts down major organ systems to divert energy to other organ systems to keep you alive. You feel as if your senses are heightened. You get this tunnel vision and you can hear everything around you, right? That's that adrenaline rush. And this is the pathway that is responsible for it, right? And it has to be this way. It's one of the reasons why we have to understand that this pathway is so complicated, because it has to be, right? We have to find a way to take one molecule of epinephrine that binds to a cell and turn it into 100 million molecules of this glucose that we can use for energy rush, that we can run away from something very quickly or fight something, okay? So that's why we see this, and that's why we've got these massive amplifications, right? This is a fine-tuning of this response. Remember I said, wouldn't it be easier if a cell could just take one molecule of epinephrine and instead of having all this crazy stuff happen inside of the cell where it's activating this, and this, wouldn't it be just easier to send one molecule with that information to where it's got to go? It'd be easier, but it would not be as efficient for your body to stay alive and do what it needs to do to stay alive, okay? Now remember that example I just talked about was in one cell. You think one cell gets activated in your adrenaline response when you see a bear? Absolutely not. You have hundreds of millions probably get activated at some point or have some kind of stake in that particular interaction, okay? So these enzyme cascades amplify that cell's response, and it needs to happen that way. Otherwise, you can't fight the bear. So at each step, the number of activated products is obviously much greater than the preceding step. We saw that in the previous example. This pyramid scheme right here is kind of a similar type of reaction where the person at the top kind of is, the thing at the top is maybe the epinephrine molecule, and the thing at the bottom is all of the different molecules that become activated or available as a result. Now, at any point during that particular pathway that we just talked about here, you can regulate it. You can shut it down. You can tone it down. You can maybe even sometimes, depending on the influx of certain types of chemicals, you can change that particular pathway and activate something totally different, okay? So the reason this has to be so complicated is because we like to regulate it, and at some point we have to be able to shut this stuff down, okay? Which we will eventually talk about, the shutting down of this particular, these particular processes as well, okay? So I just described to you in fairly great detail a cytoplasmic response. That is activation of a certain type of response in the cytoplasm. That's not something that happens in the nucleus, okay? So we'll talk real briefly here about nuclear responses as well. Not nearly as detailed because there's plenty of different nuclear responses, but most of the nuclear responses are kind of the same when you boil them down to exactly what they are, okay? Many of those other signaling pathways regulate the synthesis of enzymes or other proteins. And the only way that we can regulate the synthesis of proteins is by turning on genes on the DNA in the nucleus, right? That's what our DNA does. Our DNA, when you express the DNA, when you transcribe it into mRNA and then translate it, you translate it into a protein, okay? So we can turn genes on or off. We can turn on transcription or turn off transcription, Okay, that's the same as turning genes on and off. And so the final activated molecule may function as a transcription factor in the nucleus. So we have this massive signal transduction pathway that takes place, and maybe the very final molecule that is activated, maybe it turns out to be something called a transcription factor. We're going to talk more about transcription factors next semester, but they are vital proteins that are needed to turn on genes, okay? And we'll talk more specifically about them when we get there. So this is a diagram of how nuclear responses work, okay? We see everything that we've seen so far. We see in this case, they use a growth factor as their example instead of epinephrine, but a growth factor is going to bind here to a receptor, okay? And that receptor is going to initiate the signal transduction phosphorylation cascade, right? We talked about that in the previous video. The final protein in the phosphorylation cascade is going to turn on this protein here called a transcription factor. When the transcription factor is turned on, it binds to the DNA and it transcribes a gene, okay? It makes mRNA. And when you transcribe a gene, oftentimes you get the protein product as a result, okay? So I'm not going to spend as much time on the nuclear responses as I did on the cytoplasmic one, because the cytoplasmic one was a very specific one. And when you talk about turning on genes, you can turn on any gene, really. You can turn on just about any gene this way. And when you turn on a gene, they all kind of do the same thing. We're going to talk about gene transcription, gene translation next semester. So we will go into more detail on this. It's just something we don't need to do this chapter, okay? So I've got another animation. I'm hoping that these animations work. Again, if it doesn't work, make sure that you let me know in class and I can show you them in person, okay? So I'm going to get out of the presentation, go to the animation.
A signal transduction pathway ultimately sets in motion some cellular response, such as alteration of metabolism, rearrangement of the cytoskeleton, or modulation of gene activity. Click on glycogen to see how signal transduction might affect metabolism. Click on a microtubule to see how the cytoskeleton might be changed. Click on DNA to see how signals can affect genes.
Okay, so we'll click on glycogen first. I think that's going to get into the epinephrine pathway that I spent a little bit of extra time on. So you can kind of see that in motion here. And when you see this thing go in motion, try to relate it to some of the diagrams that we've seen in the previous videos, okay?
Here's an example of how signal transduction affects metabolism. The fight or flight hormone epinephrine causes the liver to release sugar into the blood for energy. In a liver cell, epinephrine acts by triggering a cascade of protein kinases, which eventually activate molecules of the enzyme glycogen phosphorylase. This enzyme cleaves glycogen molecules, releasing sugar in the form of glucose 1-phosphate.
Okay. So let's click on the DNA, because that's one we've talked about as well.
A protein kinase might activate a gene and trigger the synthesis of a new protein. The protein then redirects cell metabolism.
Okay, there you go. And creation of the protein right there. All right, let's do the cytoskeleton.
The cytoskeleton can be rearranged when a signal transduction pathway releases a flood of calcium ions into the cytoplasm. These ions activate a protein called calmodulin, which triggers the assembly of microtubules that alter the shape of the cell.
Okay, so a lot of this stuff, guys, that you saw in the animations is highly detailed, and there are certain aspects of it that you definitely do not have to remember. But, for instance, you don't have to know the name of calmodulin. That's not going to be something that I test you on specifically. I've already told you that the interaction of the second messengers there you saw with the turning on of the gene in the nucleus with IP3 and calcium. You don't have to know those specific not necessarily functions, but the way that they interact with each other. You don't always have to know. You don't have to know the specific names of like glycogen phosphorylase. Okay, but you do have to know that it's part of the phosphorylation cascade, the proteins that are sequentially activated. So there are certain things that you have to know in terms of all of these pathways, and other things that you don't. If you are questioning what you need to know, let me know. That's what we have class time for. Bring those questions to my attention, okay? So as you go back through the notes and as you watch these videos and rewatch them, please write down anything that might come to mind that you would like answered, right? This is kind of complicated stuff. There's a lot going on here, and I want to make sure that you have ample resources to be able to do that. Additionally, as I start to put the Unit 3 test together, if there's questions that I come across that will require you to understand more in-depth information about certain things, I will absolutely go back and review them. But you have to remember that when I do those types of things, you have to write them down and make sure that you remind yourself that I did cover those. Okay, so let's kind of put the finishing touches on this here, and let's touch on the last slide. The last and maybe one of the more important ones. Maybe not even more important ones. It's important, but it's easy to remember. We have to be able to deactivate these cell signals. Okay, there has to be some way for us to shut down the epinephrine rush. Right? Could you imagine just being your body locked in that perpetual state of epinephrine rush, of adrenaline rush? You're probably glad that your body shuts it off at some point, because sometimes you get these adrenaline rushes and you just feel exhausted afterwards. And the reason for that is because you've used a large store of your built up and stored up sugar, right? And your body has just undergone a massive exodus of ATP. It's being used to do something to either keep you alive or respond to some kind of situation, okay? So the signal transduction pathway allows for the deactivation of this, right? It allows for you to be able to shut these things off. At any point in time during that phosphorylation cascade, we know that we can shut it off. Phosphatase will shut those things off. Okay, that's the enzyme that shuts off those protein kinases. And if we remove the signal molecule, the very initial ligand signal molecule, it'll shut down the entire process as well. Okay. Now there's many different ways that those signal molecules can actually be removed. Some of them can be replaced by other things. Sometimes the receptor molecules can literally kick them out. Other times there's interactions with different parts of the receptor molecule by different molecules, which will help kick out those ligand molecules. And sometimes there's other chemicals and chemical reactions that will degrade or break down that signal molecule to remove it from that receptor as well. So there's plenty of ways that we can shut this thing off, or these things off, and if we had no way to shut them off, we'd have been dead a long time ago. Okay, so keep that in mind, that even though it seems like a simple thing, okay, we're just going to shut this thing off, it's an absolutely vital thing that you have to have a healthy respect for, because if you couldn't shut it off, you'd be dead real quick. Okay, so that'll be it for chapter 11. We're going to do some other supplemental things with chapter 11. So remember, if you have any questions, make sure you write them down, and I'd be happy to answer them. Have a good one, guys.